Hair is an appendage of skin which grows out of an organ called a hair follicle. It resists chemical decomposition and retains structural features over a long period of time. The cuticle is the outside of a hair formed by overlapping scales that point towards the tip of the hair. Imbricate, mostly found in humans. Spinous, mostly found in minks, cats, and seals. Coronal, mostly found in small rodents, bats, and sheep. The cortex is the part of a hair that is made of spindle-shaped cells aligned parallel to the length of hair embedded with pigment granules. The medulla is a collection of cells appearing as a central canal. The medullary index is the measurement of the diameter of the medulla relative to the diameter of the hair shaft. For humans, it's less than 1/3, and for animals, it's more than a half. The medulla patterns are continuous, interrupted, fragmented, and absent.
When casting a hair, you need 4 essential things: slide, tweezers, nail polish, and of course your hair. First, you want to start out by applying 1 small line of nail polish. As such. Next, take your hair and gently lie it down across the nail polish. Now, we be patient and let it sit for 5 minutes.
A few moments later.
Once your slide is on your microscope, look to find what you're looking for, and you should have your cast. The first circle is the 100x circle. This is for the yellow objective lens. When filling this one out, go to the yellow objective lens and draw what you see. This next circle is 400x. This is for the blue objective lens. This third circle is for the cast. When filling this one out, you do the same as the previous 2 and draw down what you see. These next lines are pretty self-explanatory. Sample is what sample of hair you have. Description is what it looks like if it's long, thick, or what color it is. Medullary index is what % medulla it is. The medulla pattern is for what pattern the medulla is. And finally is the cast pattern. And that's what cast pattern you see when you did the cast.
A fiber is a thread of filament from which a vegetable tissue, mineral substance, or textile is formed.
To determine what a fiber is made of, you need to burn it. So let's burn it. It's kind of hot. After you burn it, you look at the residue, and if it's melted, then most of the times it's made out of plastic. And if it caught fire, like this one just did, it has very natural occurring fibers in it, such as hair.
To the untrained eye, a crime scene can appear to have no clues. But to a crime scene investigator, or CSI, they're everywhere.
This is the largest forensic teaching centre in the UK, run by the National Police and Improvement Agency. Here students are trained to gather and preserve evidence.
And this week, I'm joining them.
A stolen car has been found abandoned after a hit-and-run incident. But it's not for real, it's a mock-up designed to put me to the test. My task is to gather hair and fibres from inside the car. I'm hoping these will help me pinpoint the thief from our volunteer suspects. Helping me is CSI tutor Paul Deering.
Hair and fibres need to be recovered very carefully because they can easily be disturbed within the scene and actually lost.
Before we start, Paul and I put on special suits so we don't contaminate the scene with our own hair and fibres. All of us leave a trail of hair and fibres wherever we go. Every day, the average person loses 70 to 100 hairs from their head alone.
There's quite a lot in here.
It's not uncommon with the velocity of the collision and the impact the head has with the airbag to find a fair amount of hairs.
Although only one of our suspects has hair of the length we've found, the others may have shaved their heads since the crime, so they can't be eliminated at this stage. Next, I collect fibres from the seat. Most items of clothing are made up of tiny fibres. Like hairs, these can easily be left behind.
The more fibres that we can recover actually makes the evidence stronger when the scientists analyse it.
Right, we've got all our evidence all properly packaged and logged. Now it's off to the forensic lab to see what it can tell us about our suspect.
Peter Grant is an expert in hair and fibre analysis. The first step is to look at our evidence under a microscope. Fibres look very distinctive when magnified, so identifying different types is straightforward. Naturally occurring vegetable fibres like cotton look flat and twisted. Animal fibres such as wool appear scaly, while synthetics look smooth and speckled. Can Peter help me identify the fibres I collected?
We got a garment that's made up of two different types of coloured fibre, but both acrylic.
So it's certainly a start, isn't it?
What we'd need to do now is to have the garment that we think the suspect may have been wearing, and then we can do a whole range of comparison tests.
One of these tests can separate the individual colours in the fibre dyes. Forensic scientists can then compare these colour fingerprints to see if they match. Next, we look at the hairs I gathered. Microscopic analysis of these can also reveal a great deal of information, such as the person's race, which part of their body the hair is from, and whether it's been dyed.
You've got a bit of a lump on the end of the hair there. What does that mean?
That's the root of the hair. This is something else we can tell about this hair, is that it's been pulled out and it's taken with it some tissue from the skin. That's the bit that we can DNA test.
Another tool available to the forensic scientist is the toxicology test. This reveals if a person has been taking drugs, because the chemicals in the human body get secreted into hair as it grows. This type of analysis was key in the case of abducted schoolgirl Shannon Matthews. When samples of her hair were analysed, various drugs were discovered, including some sedatives. This proved that Shannon had been drugged even before she was kidnapped. Back to my evidence, and with the help of Peter, I've confirmed my suspect is a white man with brown hair, wearing an acrylic garment with two colours. If these had been real suspects, I'd have found my man.
TV shows like CSI bring a touch of glamour into the world of forensic science. While we know such programmes aren't real life, the work of forensic scientists is very real and crucial in keeping one step ahead of the criminals.